So, for me, it's my pleasure to present Professor Amar Sahai. He's an associate professor at the Center for Regenerative Medicine at Mass General Hospital and the James and Audrey Foster Mass General Hospital Research Scholar. He's also an associate member of the Broad Institute of MIT and Harvard. The mission of his lab is to generate fundamental insights into the role of hippocampal circuit mechanism and network plasticity process such as adult hippocampal neurogenesis in memory, processing, and modulation of emotion. Today, Dr. Sahai will present inhibition, memory, interference, and indexing. Please, Dr. Sahai.
Good morning and buenos dias. Thanks so much, Antonello, and the other organizers here in Boston, as well as in Chile, for putting together this meeting. These are difficult times, but I think this virtual format allows for a greater access, especially for trainees to science around the world. And I think that's one of the silver linings during this time. Just by way of disclosures, some of the work I'm going to present today is the basis for a patent that was awarded to us a few years ago. So as an introduction, I think it suffices to say that the hippocampus plays a critical role in the formation of episodic memories, memories of what, when, and where. And it's thought to do so by integrating highly processed sensory information that arrives from higher order cortices and generates conjunctive representations that relate places, people, events with the spatial temporal context in which they occur. This information that's encoded in the hippocampus is then transferred to the prefrontal cortex for storage, a process that's a gradual one and it's referred to as memory consolidation. During memory consolidation, this conjunctive-rich, detail-rich representation is transformed into a more abstract, gist-like structure. And this transformation process of memories during consolidation is thought to facilitate the generation of schema that's essential for new learning. But this loss of details also results in generalization. And this may be particularly problematic for traumatic experiences. So for example, an experience in combat, a veteran returns back to civilian life, and this loss of details of this episodic representation of these experiences into more gist-like structures may result in the expression of fear in neutral contexts. So for example, this house here, which is part of the original conjunctive representation, the scene in combat, is sufficient then to evoke or retrieve the memory. And once the memory is retrieved, the hippocampus essentially releases the brakes on subcortical circuits that mediate the expression of fear and physiological symptoms like heart rate, skin conductance, blood pressure, which essentially is the pathophysiology of PTSD. And this concept of how the loss of details as conjunctive representations are transformed into more just abstract-like structures may result in generalization is illustrated in this cartoon on this particular slide. So imagine if you're sitting in a cafe, it's raining, some beautiful flowers outside, there's a green trash can, there's some bicycles, and there's a bomb blast. Now this conjunctive, detail-rich episodic memory over time during consolidation is transformed into this more semantic, abstract, gist-like structure. Naturally, it is more generalized because the statistical probability of just a trash can and a bicycle in our environment is significantly greater than all of these items that were originally bound in that conjunctive representation. And this results then in this bicycle or a trash can retrieving or evoking the retrieval of the memory, and consequently then the elevation in blood pressure and skin conductance and heart rate and such. This property of memories to be transformed and become less precise over time and lose details has been captured in numerous studies in mice and rats. And this is an example from studies in mice and rats using a contextual fear conditioning paradigm, where an animal is conditioned to a context A, that's associated with a mild foot shock. When these animals are placed back at a recent time point in either the training context or a distinct, safe, neutral context C, these animals exhibit elevated levels of freezing behavior, indicative of having made that association of the context with the mild foot shock. In contrast, these animals exhibit very low levels of freezing behavior in this neutral, distinct context C. However, with the passage of time, as this memory becomes less precise and more gist-like, when the animals are returned at a remote time point to the training context or the neutral context, you now see this elevation in freezing levels in the neutral context, indicative of increased generalization of fear. You also notice this decrement in the memory of the original training context. Over the last decade, in vivo physiological recordings in the prefrontal cortex, molecular tagging approaches, the use of immediate early genes has demonstrated that memories undergo profound reorganization in the prefrontal cortex. And in the hippocampus, the original representation begins to decay over time during this process of consolidation. But the hippocampus plays a continuous role in the retrieval of memories even once they become remote. And this is work now from many different labs that's been data has been accumulating over the last 5 years or so. However, the precise contributions of the hippocampus to the reorganization of memories in the prefrontal cortex is poorly understood, let alone how the hippocampus may dictate the extent to which memories retain details with the passage of time. In today's talk, and I should point out that our own thinking of the contribution of the hippocampus has been greatly influenced by a theory that was proposed almost 30 years ago that has only recently gained tremendous traction. And this is the hippocampal indexing theory. It was first proposed by Tyler and DeSena in 1986, and which posits that the hippocampus registers experiences in ensembles or engrams, and that these engrams ensembles function like indexes, much like an index card in the library. For everyone out there in my generation who's been to libraries, these index cards are linked to details in books distributed in shelves, much like the neocortex. And for all the millennials out there who've never been to a library, it's like a Google search word, which is linked to the details distributed in servers around the world. And the activation of that index in the hippocampus facilitates memory retrieval by virtue of reinstating the original patterns of activity in distributed sites in the neocortex, as well as, I would argue, in subcortical circuits that underlie the original encoding of the experience. A direct consequence or take home from this theory is that the integrity of the index in the hippocampus dictates the extent to which we can retrieve precise memories stored in the neocortex. We recently proposed, building on a large body of work on systems consolidation theory on the shoulders of giants, that the integrity of the index, by virtue of levels of memory interference, dictates the extent to which memories retain details in the cortex. And the idea is very simple. As we encode new memories, we generate new indexes in the hippocampus. So returning to that bomb blast example that I gave you, that registers in the form of ensembles in the hippocampus and generating an index. However, with new experiences, there is interference between old and new indexes in the hippocampus. And this determines the extent to which old indexes retain their integrity or persist. We argue that this level of interference dictates the extent to which the cognate cortical representation for that index maintains details. So the corollary is that by reducing interference between new and old indexes in the hippocampus, or by preserving the index in the hippocampus, by maintaining its integrity, we may facilitate the retention of details that conjunctive representation that is stored in the neocortex and that is associated with that index in the hippocampus. In today's talk, I'm going to share with you our insights over the last 8 years, generated from in my lab over the last 8 years, on an inhibitory microcircuit in the dentate gyrus CA3 region of the hippocampus, and how this inhibitory microcircuit contributes to memory consolidation and time-dependent generalization. Additionally, we will look at how this inhibitory microcircuit causally affects the evolution of ensembles in hippocampal cortical networks during memory consolidation. And we will look at this data through the lens of hippocampal memory indexing and interference. So this is the circuit, the microcircuit of interest. It's the granule cell parvalbumin inhibitory neuron CA3-CA2 microcircuit. As many of you know, and I've heard from talks earlier today and last few days, granule cells have axons called mossy fibers that release glutamate onto thorny excrescence-like spines of CA3 neurons and CA2 principal neurons. CA2 principal neurons don't have thorny excrescence-like spines. It's a characteristic of CA3 neurons. Nevertheless, these mossy fibers also have these filopodia that express VGLUT1 and that release glutamate onto parvalbumin inhibitory neurons in stratum lucidum. So when you stimulate a single granule cell, you simultaneously recruit feed-forward inhibition mediated by these parvalbumin inhibitory neurons onto CA3 and CA2, but also feed-forward excitation through the mossy fiber CA3-CA2 synapse. And it's been theorized that this balance of feed-forward excitation and feed-forward inhibition dictates the extent of burst firing of CA3 neurons and the sparseness of activity in that network, which one would predict influences the extent to which memories may be retrieved in the highly recurrent collaterals of CA3. When I started my lab in September 2011, we were inspired by a study that came out from Pico Caroni's lab, I think several months prior, that had made a link between this granule cell parvalbumin inhibitory neuron connectivity, the substrate for feed-forward inhibition, and the generalization of memories. And this is work by Sarah Rudiger when she was a graduate student in Pico Caroni's lab. So returning to this paradigm where animals are conditioned to a context and then returned to that training context or a neutral context, where you see this generalization of fear in the neutral context at the remote time point, what Sarah Rudiger and colleagues found was that two things. When animals perform a hippocampal dependent task, you get an increase in granule cell parvalbumin inhibitory neuron connectivity through these mossy fiber terminal filopodia. But two, as the memory generalizes over time, you see this decrease in mossy fiber terminal filopodial connectivity with PV cells. So we set out to identify molecular regulators of granule cell PV neuron connectivity. And this is work that was driven by a very talented postdoc in my lab, Nanan Guo, who now has her own lab in China. And what we found was this gene ABLM3 that is expressed only in granule cells of the dentate gyrus. But if you zoom in, you find that the protein is only found in the mossy fibers. And if you further zoom in, you'll see that ABLM3 co-localizes with these Z01 positive puncta adherence junctions. And it's absent from the active zones that are labeled by Bassoon. These puncta adherence junctions are thought to stabilize the mossy fiber terminal on the dendritic shaft of these principal neurons in CA3. What Nanan found was that in a contextual fear conditioning task, we see a downregulation of ABLM3 protein in these mossy fibers in the hippocampus. And through a series of experiments that are documented in this paper, we found that ABLM3 levels that naturally go down with learning, if you artificially drive levels down, akin to what is seen with learning, you get an increase in mossy fiber terminal filopodial contacts with parvalbumin inhibitory neurons. And in turn, these parvalbumin inhibitory neurons, in response to increased granule cell inputs, elaborate their inhibitory contacts onto CA3 and CA2, as shown over here. Thus, ABLM3 acts as a brake on feed-forward inhibition, a learning-dependent brake on feed-forward inhibition in the dentate gyrus CA3 circuit, and dictates the extent to which granule cells recruit PV inhibitory neurons in stratum lucidum, which in turn determine the extent of blanket and perisomatic inhibition exerted onto the CA3 and CA2 principal neurons. These anatomical changes that are schematized in here on this slide were corroborated with physiological recordings in CA3 neurons, as shown over here, as well as in PV neurons, and you can find that in the paper. Basically, in this experiment, we expressed channel rhodopsin in the dentate gyrus. We either downregulated ABLM3 with a shRNA or a control non-target RNA, and then we recorded evoked inhibitory and excitatory postsynaptic responses in CA3. And what we found was that in animals in which we had increased feed-forward inhibition connectivity, we saw, as predicted, a decrease in the EI ratio, which was driven by an increase in the evoked inhibitory postsynaptic responses in CA3 neurons. Together with a larger body of physiological analysis ex vivo and slices, these data suggest that ABLM3 acts as a brake on feed-forward inhibition in the dentate gyrus CA3 circuit, and its downregulation of this shRNA in granule cells enhances feed-forward inhibition onto CA3 and CA2. So what is the effect then on generalization of memories over time? And so we return to the same behavioral paradigm that I've showed you, where we injected now shRNA or non-target shRNA viruses in the dentate gyrus of mice, and conditioned them to a context A with mild foot shocks, and then return them at the recent time point to the training context A or a distinct neutral context C, and then did the same thing at a remote time point at day 16. And what Nanan found was that these animals behaved equivalently in terms of associating the context with the mild foot shock, and as well as levels of generalization at the recent time point as captured by levels of freezing behavior in the neutral context C. However, at the remote time point, what we found was that the animals in which we had increased feed-forward inhibition now exhibited greater memory consolidation at the level of behavior, as evidenced by higher levels of freezing in context A, and reduced time-dependent generalization as captured by reduced levels of freezing in the neutral context C. So on the basis of these observations at the level of behavior, we then asked, based on our model, are we in some ways influencing the persistence or the perdurance of the index in the hippocampus, such that the animals in which we've increased feed-forward inhibition retain the details of that original experience of context A? And this is shown over here. So does decrease in ABLM3, which is increase in feed-forward inhibition, maintain the index of context A? To do that, we first asked, well, does the engram of context A show any differential connectivity with parvalbumin inhibitory neurons? And for that, we leveraged these molecular ensemble tagging tools, first engineered by Leon Reijmers and Mark Mayford in 2007. And we used the CFOS TTA mouse line that allows us to capture or tag ensembles in response to a discrete experience, in this case, a context A associated with the foot shock. And then we use a reporter mouse line, TETO tau lac Z, to visualize the connectivity of those cells that were participant in the ensemble. And what Nanan found was that the engram-bearing cells in the dentate gyrus exhibited greater connectivity with parvalbumin inhibitory neurons relative to non-engram-bearing dentate granule cells. So that is the first interesting take-home message. And the other was that as the memory generalized, we found that there was a decay or loss of this preference in connectivity of all granule cells with parvalbumin inhibitory neurons. So this motivated us to ask, well, if we now artificially increase the connectivity between granule cells and parvalbumin inhibitory neurons, can we prolong or maintain the engram or the index in the hippocampus? And to test that, we now use the CFOS TTA system in combination with a TET-O channel-rhodopsin reporter delivered virally. In this instance, we can now tag the ensemble in response to context A association with the foot shock with channel-rhodopsin. And this allows us now to artificially activate the ensemble of context A at recent and remote time points and see if we can access the memory. In one group of animals, we increased feed-forward inhibition, and the other group of animals received the non-target shRNA, the controls. Consistent with prior work, we found that at the recent time point, at day 2, in both groups of animals, when you stimulated the ensemble of context A, when the animals were placed in a very different context, context C, the animals now expressed high levels of freezing, suggestive of retrieval of that memory of context A. And this has been replicated by many different labs and was first demonstrated by Liu Ramirez and Susumu Tonegawa. But the interesting question for us was that at the remote time point, can we still access the memory? And to do that, we then stimulated the ensemble of context A, or the engram of context A, in the dentate gyrus at day 10. Unlike in controls, where we could no longer access the memory, and these animals showed low levels of freezing, even following stimulation of channel-rhodopsin with blue light, the animals in which we'd increased feed-forward inhibition increased levels of freezing in the neutral context, indicating or suggesting that we were still able to access the memory of context A, and that increasing feed-forward inhibition in the dentate gyrus CA3 circuit extended or prolonged the maintenance of the hippocampal index. So we next asked, how does maintenance of the index in the hippocampus affect the reactivation of memory traces in hippocampal cortical networks and subcortical networks with the passage of time? And to do that, we now combined the CFOS TTA system with a TETO H2B M-Cherry reporter line. And what's special about H2B M-Cherry is that it's taken up by histones, and so consequently it permanently labels the cells that were originally tagged. This permanent labeling allows us now to assess if the representation, or let's say the ensemble or the engram of memory of context A is reactivated when the animals are placed in the neutral context C at the recent time point or at the remote time point. And we can do this by combining H2B M-Cherry with CFOS immunohistochemistry. And that tells us if the original ensemble of context A is reactivated when the animals are placed in context C at the recent or the remote time point. What Nanan found was that if you look over here, that in CA3, CA1, and to our surprise in the anterior cingulate cortex, in the animals in which we had increased feed-forward inhibition in the dentate gyrus CA3 circuit, we reduced or decreased the reactivation of the ensemble of context A when the animals were placed in context C at day 16. So this gives us a sense of how we think reactivation of the ensemble is driving this this freezing behavior in the neutral context. When the animals are placed in the neutral context at this remote time point and they exhibit high levels of freezing, it's because they're retrieving and aberrantly retrieving the original context A ensemble in CA3, CA1, and in the ACC. But if you increase feed-forward inhibition, you constrain this aberrant reactivation, and you decrease at the level of behavior freezing levels in neutral context C. This was also, to our surprise, this was also seen at the level of the basolateral amygdala. So feed-forward inhibition in the dentate gyrus CA3 circuit maintains the engram, or we think the index, in the hippocampus, and this in turn dictates the extent to which distributed traces in the cortex as well as in subcortical circuits like the BLA are reactivated at these remote time points. So in as much as these molecular tagging approaches are very powerful, hopefully you can appreciate from the data that I've presented in so far, they also have their limitations. So for example, we can only infer about the properties of the ensembles, the properties of the participant or the constituent neurons. We have no idea about how the ensemble evolves over time. We have no idea about the synchronicity or the firing of patterns of the constituent neurons in those ensembles. We can only infer based on these molecular tagging approaches. And so to get a better handle of how feed-forward inhibition in the dentate gyrus CA3 circuit influences the evolution of these ensembles in hippocampal cortical networks over time and influences the properties, the firing properties, the neural activity of these constituent neurons in these ensembles over time. Hannah Twierkowski, another very talented postdoc in the lab, along with Victor Steininger and Min-Jae Kim, developed, optimized, and implemented a pipeline that allows us to perform in vivo calcium imaging longitudinally in the same animals in CA1 or in ACC, in animals in which we can increase feed-forward inhibition in underlying upstream DGCA3. And she used the same behavioral paradigm that I've shown you all along, where animals are conditioned to a context, context A, they then are returned to context A or a neutral context C at the recent time point, and then again, as the memory is consolidated at this remote time point. So this is just an example at the level of the anterior cingulate cortex, representative traces from a few neurons as the animal is returned to the training context. So Hannah now was able to image over time and look directly at the evolution of properties of these ensembles, whether they fire at different frequencies in terms of calcium transients, their participation in ensemble-like activity as assessed by synchronous activation in CA1, as well as in anterior cingulate cortex. So when Hannah took these animals and returned them following fear conditioning a day later, so on day 1, to the training context or the neutral context, she found, consistent with our prior work, that both groups of animals exhibited high levels of discrimination between the training context and this neutral context, context C. She then zoomed in and looked at the activity of neurons in CA1 and ACC in animals in which we'd either increased feed-forward inhibition or controls. And what she found was that in both groups of mice, at the level of CA1, we see the emergence of a learning-induced ensemble that's associated with the training context, as shown over here, and as conveyed by correlated pairs of neurons or synchronous activity of neurons in CA1. To our surprise, though, we found during these early stages of consolidation in animals in which we had increased feed-forward inhibition in dentate gyrus CA3, we saw the emergence of a training context-associated ensemble also at the level of the ACC, suggestive of increased hippocampal cortical communication in the animals in which we had increased feed-forward inhibition in dentate gyrus CA3. Hannah then inquired into or examined the specificity of the ensembles, and she looked at these neurons, what we call A and C neurons, which are the neurons that are active in both context A as well as in context C, as shown over here. And what we found was that in animals in which we had increased feed-forward inhibition in dentate gyrus CA3, we saw a reduction in the number of these A and C neurons in CA1. These data are schematized over here. Looking at CA1, both groups of animals exhibit a learning-induced ensemble that's associated with the training context. However, in the animals in which we have increased feed-forward inhibition in dentate gyrus CA3, we now see the emergence of a training context-associated ensemble also at the level of the anterior cingulate cortex. We then asked, how do these ensembles change over time? And I should point out that at the recent time point, we also found, by looking at these A and C neurons, that in animals in which we had increased feed-forward inhibition in dentate gyrus CA3, we saw a decrease in the number of A and C neurons at the level of CA1, indicative of greater specificity of that ensemble. So we then asked at the remote time point, when memories become less precise, what is the effect of feed-forward inhibition on the evolution or the arc of evolution of these ensembles in CA1 ACC networks? First, Hannah assessed at the level of behavior, and what she found was that the animals in which we had increased feed-forward inhibition in dentate gyrus CA3, the shRNA group, exhibited higher levels of discrimination than controls. And this was driven by an increased level of freezing in context A relative to context C. She then zoomed in to CA1 and ACC, and now what she found, so within the same animals longitudinally over time, one, in animals in which we had increased feed-forward inhibition in dentate gyrus CA3, we saw at this remote time point the maintenance of a context-associated ensemble in CA1, as well as the emergence of the neutral context, context C, associated ensemble in these animals, as shown over here. We then asked if there are any differences in these A and C neurons. And what Hannah found was that at the level now of the anterior cingulate cortex, this was not seen at the recent time point, we saw a reduction in the number of neurons that are active in both A and in C, but only in the animals in which we had increased feed-forward inhibition. So if we compared the shRNA group with the controls, we saw that the shRNA group had fewer A and C neurons, so neurons that were active in both contexts at the level of the anterior cingulate cortex. So these data are schematized over here for the remote time point. We find that there is the persistence of a context-associated ensemble in CA1 over time in the animals in which we have increased feed-forward inhibition. So we saw it as early as the recent time point, and we see it also at the remote time point, as well as we now see at the level of the anterior cingulate cortex a reduction in the number of these A and C neurons in animals in which we had increased feed-forward inhibition. Finally, Hannah trained a classifier on these A and C neurons. These A and C neurons also show a difference in level of activity, and that was used to train the classifier. And what she found was that training the classifier on the activity of A and C neurons in CA1 at the remote time point resulted in greater prediction accuracy of context relative to the controls. So in conclusion, these data begin to highlight how an inhibitory microcircuit at the level of dentate gyrus CA3 contributes to the consolidation of memories in hippocampal cortical networks and generalization of these memories. And our work fits into a larger body of work that has implicated parvalbumin inhibitory neurons in CA1 and in ACC. So this is work by Paul Franklin's group and Sarah Aton's group, showing that these parvalbumin inhibitory neurons drive or mediate or regulate or engender synchronous activity of principal neurons in CA1 and ACC and facilitate hippocampal cortical communication, as was shown by Paul Franklin's group, specifically looking at coupling of sharp wave ripples and spindles in the PFC. From the perspective of hippocampal indexing theory and memory interference, our data suggests that this inhibitory microcircuit, the granule cells recruitment of parvalbumin inhibitory neurons, and provision of perisomatic inhibition onto the principal neurons in CA3 facilitates the stabilization of the engram, which we think now is functioning like an index, to then dictate the extent to which memories retain details in prefrontal cortical networks. Our work provides more evidence for the transformative account of multiple trace theory and indexing theory. And there are several predictions that come out from our work. Based on the data, a lot of which actually I haven't shown you here, we would predict that at a molecular level, factors that destabilize branched F-actin and mossy fiber terminals would promote granule cell connectivity with parvalbumin inhibitory neurons. A direct outcome, a prediction is that feed-forward inhibition and DGCA3 promotes hippocampal cortical communication based on the data that I've shown you. That's probably mediated by an elevation in frequency or duration of ripples, as well as coupling, perhaps, between ripples and spindles in the PFC. And these are experiments that we are actively pursuing. One other prediction is that if you inefficiently consolidate a memory, it is more likely to be generalized over time. And this data should actually be available from studies in humans as well as in rodents. And so the failure of trials that include that test drugs like propranolol that may not efficiently block consolidation, you may get this increased generalization. So you either want to erase the memory trace, or you want to completely consolidate the memory efficiently. But anywhere in between, the prediction is that you would increase generalization over time. And then finally, something that we've been thinking about a lot is that there is a trade-off. During consolidation, the generation of GIST abstract representations is essential for the generation of schema. And schema is how we perform or we learn new things. We would predict that there is a trade-off as these representations lose details. So our manipulation that preserves the representation in terms of its details may result in inefficient generation of schema that's important for new learning. And so both at the level of the circuit, we'd like to understand how schema are generated, as well as how this balance might be toggled by upstream mechanisms that reside in the hippocampus. Finally, I want to thank all the folks who've done this work. These projects were driven by a very talented postdoc, Nanan Guo, who has her own lab in China, and another very talented postdoc, Hannah Torkowski, who will hopefully go on the job market in the next year, or maybe even a little bit earlier, based on this work and other work that she's doing in the lab. I'd like to thank funding agencies for their support. I'd like to thank my collaborator, Larry Zweifel, who contributed to the first half of this story. And then last but not least, I would be remiss if I did not thank all the first responders, at MGH, all the hospitals in America, in South America, in Asia, in Africa, because I think it's because of their tireless efforts that many of us can return to some sense of normalcy, continue our science. And so I am forever grateful to all of these people around the world who have, contributed to battling this pandemic that's ongoing. So thank you very much, and I'll be happy to take questions.
Okay, thank you so much, Amar, for this great presentation. It's so interesting. So for the public or please send the question by the Q&A application or preguntas y respuestas. There are a question, Amar, from Andres Rojas. Hi, knowing that emotional cues are important for generalization of traumas, how the input from emotional neuron circuits as amygdalas or nucleo accumbens is incorporating this memory circuit generation. Is there an effect of modulation in the feed-forward inhibition?
Yeah, it's a great question. So, and there are several ways, again, these aren't, the conclusions are presented aren't by any means final. It's all work in progress based on our work and other labs. But now we can begin to think about how certain memories do retain details. And in particular, as you pointed out, emotional memories. And so based on these studies, we would predict that noradrenergic inputs, other inputs that convey salience to the hippocampus may ensure the preservation of that index in the hippocampus and the extent to which details of that memory are retained in the cognate cortical representations. This could also happen in between the hippocampus and the cortex. It could happen at the level of local microcircuits, at the level of the PFC. But now we have the tools to begin to think about it. It could happen at the level of CA1 and stabilization of the ensemble in CA1, because we think CA1 plays an instructor, like teacher-like function in terms of governing the reorganization of memories in the PFC. So in other words, stepping back, it could be inputs onto granule cell PV neurons that stabilize the recruitment of these PV neurons and consequently downstream CA3, CA2 neurons and maintain the index. It could be at the level of CA1 to prolong or maintain synchronous activity of the underlying that ensemble. It could be at the level of hippocampal cortical synchrony through network oscillations, which we are now doing. We don't have any evidence as yet. Or it could be at the level of local circuitry at the level of PFC to slow down that transition from a detail-rich representation of the PFC to a schema which is made up of maybe a smaller number of neurons, actually much like those A and C neurons.
Okay, thank you. And there are another question from the panelist or participant. I have a question, Amar. So, feed-forward inhibition from your experiments showed that it's very relevant to modulate this system. And how is modulated this feed-forward inhibition in endogenous?
Yeah, so we know, for example, and in our own lab, we now have evidence. Right now, I presented data that's all aversive learning. We now have evidence in the lab that appetitive learning also promotes granule cell recruitment of parvalbumin inhibitory neurons. We know from work from Pico Coroni previously that other forms of learning that also require the hippocampus also facilitate granule cell recruitment of PV neurons. So we think that this is a general substrate for hippocampus-dependent learning. And so the consolidation of memories requires the recruitment of these stratum lucidum PV cells to then dictate principal neuron activity. And then once you have synchronous activity of principal neurons, you can then generate, higher frequency firing and bursts and ripples and such. But at the level of the circuit, we know that these PV cells are critical through perisomatic inhibition and regulation of firing of principal neurons. And so we think that this is a general mechanism for memory consolidation at the level of the hippocampus. Does that answer your question?
Yes, of course. And also just for speculate, we know that post-traumatic disorder is a very relevant pathology. So you can speculate how works this circuit in that disease, in post-traumatic stress disorder.
Yeah, so what I was saying earlier was that, we would predict that contrary to what might be intuitive, one would think that you don't really want to remember the details of a traumatic experience. And often actually it is the case that people blank out when it comes to retrieving all the details. But we would predict that if you were actually able to retrieve all the details and maintain that representation of the traumatic experience, then that would result in less generalization over time. Because we think, as I said earlier, that we think one of the reasons for generalization is because the memories lose details and become less conjunctive and more abstract and gist-like. But you need that for new learning and for the generation of schema. I think the brain is biased towards generation of schema because that's essential for all new learning. And we need scaffolds to plug in information in new tasks. But the trade-off, we also know that, I mean, all of us also remember some memories in details, but we don't remember all memories. We don't remember all memories with as much detail. And we think that those few memories that we remember with tremendous detail, perhaps there is an index that's persistent in the hippocampus, or the extent to which that memory has become GIS-like is reduced at the level of the PFC.
Okay, perfect. Thank you. So I don't see more questions. So I think that is that is all. So thank you so much, Amar, for this amazing presentation. And stay for the next talk. Please stay. So thank you.
Thanks so much. Thank you, everybody. Stay well. Thanks, Antonella Antonello and the organizers.
Okay. Thank you for accepting our invitation. See you
Bye.
In the future. Thanks. So next presentation is from Pablo. Hi, Pablo. How are you?
Hi, everyone.
Hey. So we don't test the to share a screen. If you want to share, just
I'm going right away.
Just to test. Okay, looks fine.
Okay, excellent. I'd like to thank the organizers.
So
go ahead. Go ahead.
Yeah.
Go ahead. Sorry.
I just want to know that the presentation is fine to share. So I will present Professor Pablo Moya. He's a full professor from Institute of Physiology at University de Valparaíso, Chile. And also belongs to the Centro Interdisciplinario Neurociencia de Valparaíso. And he is biochemistry and interested in the central nervous system disorder using neuropharmacological and genetics approach. The main focus of his research is to study serotonin neurotransmitter system and its role in the anxiety and mood disorder. And today, Pablo Moya will present us a glutamate transporter EAT3 confers resilience to chronic stress in mice. Please, Pablo, to start the presentation, and thank you for our invitation.
Thank you, Antonello, for the introduction. Thank you to the organizers for inviting me to this fantastic, gathering of researchers, worldwide. I have to deeply apologize, to Amar and the previous speakers, because considering the lockdown and having, kids, below 2 years old at home, productivity is, quite complicated. So I was unable, to connect earlier. As Antonello pointed, my laboratory is interested, in neurotransmission, what goes awry, in neurotransmission, in mood and anxiety disorders. And although my favorite neurotransmitter is serotonin, I have been working during the last years, on a different neurotransmitter system, and particularly in a somehow historically neglected glutamate transporter among the family of them, which is the glutamate transporter E3. So I would like to, I will do my best to convince you, that this is, a potentially very cool novel target in neuropsychiatry. So given that, I was requested to provide a title, in a fast manner, I take in, the advantage that I have, the main role here, and that's essentially, where my lab is located, in the marvelous Bay of Valparaíso, that I expect, the international speakers, have the possibility to visit, in near future when Neurosur joins, and goes back, to a presential attendance. So as I mentioned, the title, the purpose of the presentation, is going to be slightly different, presenting as a novel target, in neuropsychiatric disorders in general. Considering the audience, I think there is no need, to convince you that the burden of the neuropsychiatric disorders, is, really, really dramatic, and heavily impacts, the health systems all over the world. And in this, based on this situation and the lack of fully useful and pharmacotherapies, that currently work and for the entire affected individual population, there is an urgency, on developing novel targets to improve alternative and improve pharmacotherapies. This talk is going to focus in one of the neurotransmitter hypotheses that appears on several pathologies, which is the glutamatergic transmission. We know, the historic, contribution of the serotonin hypothesis and the dopaminergic hypothesis, but here we're going to focus, on glutamine. And this is a very basic representation of a glutamatergic synapse, where we have, the vesicles, the glutamate release in red dots. We can observe the ionotropic, metabotropic receptors, and in general, the high excitotoxicity that is triggered by glutamate, the excess of glutamate, must be tightly controlled, temporally and spatially, and that's what the glutamate transporters do. They participate in the glutamate clearance, and we have learned that the biggest contribution for the glutamate clearance is due to the activity of the excitatory amino acid transporters located in the glia, for the glutamate transporters in the glia. However, there is a lesser, a lesser considered participant here, which is the transporter, of this talk, which is the E3, also known as the excitatory amino acid carrier one in another nomenclature. But this one is a little different to what we currently know about neurotransmitter transporters. In the case of monoamine transporters, they are located presynaptically, so it is obvious that they participate, in the reuptake and recycling of the neurotransmitters. In the case of the glutamate, the books, historically, told us that the glutamate is taken out by the glia, and then you have the glutamine, cycle, for the recycling of the transmitter. But in this case, the E3 is located postsynaptic in neurons. So the contribution is not that obvious, and when the estimations and the calculations about how much glutamate is being transported between IT1 versus IT2 and IT3, the glial ones do 90% of the job. And that's perhaps, why from a neurochemical perspective, IT3, was historically neglected. However, its localization, places these transporters, as a key regulator of the glutamate spillover. The degree of also regulates the degree of the perisynaptic activation, the ratio between the perisynaptic versus synaptic activation, and so on. In addition, it is important to keep in mind that this family of transporters are not exclusively glutamate transporters, because they transport, as its name says, excitatory amino acid that includes glutamate, cysteine, and aspartate. In the case of IT3, it has a wide distribution in brain, particularly in cortex, striatum, hippocampus, providing an anatomical reason to explore potential consequences of altered expression in pathologies. And why did I say this? Because
Hola.
Hola, sí.
Perdió un poco la conexión.
Sí. Bueno, para la audiencia, como se dijo ayer y anteayer, sin estas caídas no sería una videoconferencia. Ya estamos habituados a aquello, así que vamos a tratar de hacer la reconexión, por favor. Un momento. Sorry, Pablo. So the connection was down.
Oh.
Okay,
So you missed the best part. I'm going away.
Yes, yes. I think so.
Sorry about that. So we were here.
Yes, but
Okay.
the last concept about obsessive compulsive disorder, schizophrenia, was lost.
Okay. Okay. Thank you. So long story short, E3 expression has been reported to be altered in a variety of psychiatric disorders, ranging from OCD, schizophrenia, bipolar disorder, depression, Alzheimer's. So we want to explore what are the consequences of altering the E3 expression in behavioral domains that are relevant in a translational approach, to these disorders. In a very RDoc, base, we won't keep talking about the disorders, but rather about the domains. The E3 knockout mice were available in late 90s, and there were no evident phenotype related to any psychiatric disorders. The main findings were related to the lack of cysteine transport, and as you know, cysteine is the precursor of glutathione. So there was impairments in terms of generation, in neurodegeneration in aged mice related, to the lack of redox buffering capacity, generally speaking. But nothing related, to cognitive domains, depression, anxiety, compulsivity. Nothing was evident, by those years in the knockout mice. Based upon human genetics findings that we obtained, almost a decade ago, when I was doing my postdoc in the US, we found, particularly in an OCD case control study, we found variants that were associated with increased expression, and that was the original reason why we focused, parallel avenues for the generation of mouse models to manipulate E3 expression: the conditional knockout mice and the conditional overexpressing mice. And I'm going to show you here data for one of the avenues, the conditional increase E3 expression and the consequences, at behavioral functional level. So this mouse was generated by a CRE-LOX technology. This is the validation that the E3 expression is, you need CRE, to express it. The glutamate uptake, goes, up only in the presence of the CRE recombinase. We use the GFP as the stop signal, so we can genotype the mice, in a UV transilluminator, when they are pups. And these mice underwent a classical neurological and motor assessment, and we recently published, this work. So, again, this is just from a historical perspective. We're going to talk a bit, about the relevance of this mice or this model, for OCD. And why OCD? Because there is a glutamatergic hypothesis, and in the case of OCD, it comes mainly from imaging findings, from functional and structural imaging findings, in the late 90s, where they pointed to alterations in the known corticostriatal thalamocortical loop, in brain, and particularly the findings pointed toward an increased corticostriatal activity in OCD-afflicted individuals. Those data were very relevant because they also demonstrated that effective pharmacotherapy or combined pharmacotherapy and behavioral therapy was able to reduce the increased corticostriatal overactivity, in the patients who responded to the treatment. They were, very highly relevant. Other data we have pointing toward glutamate in OCD are alterations in glutamate and glutamate metabolites in CSF, from OCD probants, and some open label trials, very small, but in OCD individuals that are refractory to the common pharmacotherapy, which is SRI. Do you still hear me? Okay.
We lost you for a moment, Pablo, but we're...
Yes, you are come back.
All right, okay. So that's essentially the scheme of the corticostriatal thalamocortical loop with the parallel direct and indirect pathways where there are glutamatergic connections, in there. And in case of the genetic findings, from human studies, the gene encoding E3 is one of the most highly associated, in case control studies. However, did not reach, genome-wide significance, so far in 2 large studies. But as a candidate, it's still there. Not as strong as we wish, but there are several indications that something. And in particular, the studies, we did in 2009, where we found, SNPs that are associated with increased expression. So then we decided, having the mice, we overexpress, E3 under a pan forebrain driver, as CAM kinase 3, because most of the synapses that are receiving glutamatergic input, have that post synapse, CAM kinase 2 alpha. This was like a broad approach, and we are in parallel, testing, restricted overexpression, within, specific regions. But I'm going to show you the data, from the CAM kinase overexpression. We demonstrated, the increased activity. As in the light dark. I just received. Did I freeze?
Pablo,
No.
sorry.
Yes.
Sorry, Pablo. The last part of the previous slides was lost again.
Okay.
If you come back.
Okay. This is now. So we crossed the conditional transgenic mice with CAM kinase 3, to increase expression, in forebrain principal neurons, in general. So we demonstrated, the increase of expression, in a variety of regions, and protein, messenger RNA, function, etc. So now we go to the behavior. Anxiety. So we tested in 2 paradigms: the opium field test, the light dark emergence test, very classic. And in all the parameters, such as the time and center and number of entries in the open field, we observe that the overexpressing E3 mice, has an anxious-like phenotype. The same happens in the light dark paradigms. They take longer, to go out, to the bright illuminated area, and they spend less time there. When we administered SRIs, serotonin reuptake inhibitors, which is the only pharmacotherapy available for the OCD realm, we observed that the chronic but the not acute administration, restored the phenotype. The same happened in the light dark paradigm. This work, was done by Claudia Delgado as the master thesis, as her master thesis, a few years ago. Now we turn to the compulsive-like behavior. So we tested, marble bearing. We observed, an increased parameter, in the overexpressing mice that again was restored by chronic but not acute SRI administration. Then we assess, during the along this domain, the grooming behavior. and analyzing, as a whole, as a total grooming behavior within an hour, we observe, increased grooming, that's also alleviated, by SRIs. And recently, we dissected, the microstructure of the grooming, because this is a behavior that it has a process. It is not, disorganized. It's highly organized, and it's very sensitive to stressors. So you can dissect, this behavior, as in different phases, as highlighted here. And when we do that, we observe that compared to the overexpressing E3 mice, they perform grooming, but they do not finish, the behavior. They got stuck, in the initial phases. And this is very striking, because it's essentially what happens, in mice when you administer amphetamine. This is like an amphetamine-like grooming behavior. They initiate, the behavior, but then they do not finish. So they get stuck in phase 1 or phase 2. And in addition, these mice are unsensitive, to the administration of amphetamine to elevate the grooming. It's like a ceiling effect, in this case, even though they do respond to amphetamine at locomotor level. These are part of the results that are currently under review. Okay, so some of the audience people might be asking you, where is something about memory here, in this talk. So we analyze it, and based on the contribution of the cortical striatal transmission to the OCD relevant behaviors, we focus, our initial analysis in this model, in the corticostriatal synapse. This is a beautiful work done in the Andres Chavez lab, by Sebastian Stein. And very briefly, what I can tell you that we did not observe alterations in the spontaneous, excitatory postsynaptic currents, no changes in the minis, no changes in the PPR, and no alterations, in a short-term plasticity, protocol. And collectively, this data, suggests that there are no changes, at the presynaptic level in the corticostriatal synapse, of the E3 overexpressing mice, which is okay with our prediction, because, as I mentioned E3, is located postsynaptically. So now, okay, we switch, the assessment toward what happens at the post. And here we initially, observe, an alteration, in the decay of the postsynaptic current, and a pharmacological characterization. We found that the excitatory postsynaptic currents, are more sensitive, to the blockade by this compound, which is a NR2B subunit containing NMDA antagonist, meaning that the contribution of the current, the contribution of the 2B subunit, is larger, because they are more sensitive, to this blocker. Molecular assessment, of a series of glutamatergic, proteins revealed that the NR2A to NR2B ratio, was altered, and this was because the NR2A subunit, was dramatically decreased compared to the control group, which essentially explains, the higher contribution of the 2B, at the synaptic level. And based upon, these findings, we evaluated, what happened on long-term potentiation protocol, and we demonstrated that the corticostriatal synapses, the E3 overexpressing mice, loses, the plasticity, the glutamate-dependent plasticity, the classic, as compared, to the control mice here. You can see it here, in the white, which does not happen, for the endocannabinoid-dependent, plasticity. So something goes wrong, with the NMDA receptors that are involved, in long-term plasticity, and we are, currently, further investigated, because this is just actually a picture, at a given age, at 2 months old, what's going on. we also analyze fear memory in collaboration with Andrew Holmes, at NIH. This is the classical paradigm of conditioning. We 3 tones, unpairing and shock. There are no difference between controls and the overexpressing mice. No changes in the extinction protocol. When you perform the retrieval after, the extinction protocol that involves, the 50 tones, the retrieval is 1 day after the extinction protocol, you observe like a slight, alteration in the overexpressing mice that they tend to, do more freezing. However, when you perform the same protocol, but 2 weeks later, which is known as the spontaneous recovery, you can observe that at the very first tone, the overexpressing mice, they dramatically, freeze much more. So this indicates that the E3 overexpressing mice, have deficits in the long-term, fear extinction, which, for the people involved in the research, they say, hey, but this has nothing to do, with the corticostriatal synapses. Yes, it's ventromedial prefrontal cortex, chatting with the BLA. But this, in particular, the impairments in the long-term fear extinction, have been also reported, in OCD afflicted individuals. Now we're going to switch, a bit to another subject, of study, in my lab, which is, depression mouse models. And essentially, the 2 lines of research, interconnected, at some point, thanks to this mice. The glutamatergic hypothesis of depression, in general, we can summarize it, like the decades of investigation, that we know that the stress, elevates the glucocorticoids, and that induces, increases in the glutamate release, in many areas. Indeed, the glutamate levels, are known to be elevated in the brain of patients with depression, as well, in several animal models, of depressive like behaviors. Alterations in the NMDA and AMPA subunit receptor expression, has been reported, in major depressive disorder patients, as well as in animal models in general. And the circuitry involved in both emotional processing, in memory formation, the limbic circuit, as well as the reward system, and that are implicated, in some of the impairments in hedonic process that are linked, to depression, are they use several neurotransmitters, including, glutamate among them. In addition, it is well known, and this is not presented here, the research on fast-acting antidepressants, has also, focused, in glutamate neurotransmission, given, the impressive facts on the use of ketamine, on the research on ketamine, as a fast-acting antidepressant, in 2006, at the NIMH. Originally. So, in terms of the E3, what the literature says, in several chronic stress animal models, decreased glutamate uptake and reduced expression of the glutamate transporters, have been observed, and among them, we have E3 as well. There are 2 or 3 articles, dealing or providing in data on this. And very, very recently, last year, a revival on E3 and its role in depression, was published when the group by King and colleagues, reported that the mouse lacking E3 exhibited some depressive-like behaviors, at baseline conditions. We don't know what happens when you stress the mice in general. So since we have, the overexpressing mice, one of the students in my lab, as a side project, embrace the characterization of the impact of unpredictable chronic mild stress, UCMS, very robust behavioral paradigm, to induce depression-like behaviors in mice, in our mouse model. We perform the 5 weeks protocol with twice stressors, per day, randomly assigned. So essentially, the mice don't know what's coming, which is, one of the most realistic, protocols, if you want to consider it. And then they went through a series, a behavioral battery, assessing anxiety, anhedonia, depressive-like behaviors, and a bit of memory impairments, memory test. So this is a marvelous work done by Nicolas in the lab. He recently, defended, his PhD thesis. He first, established here in Valparaiso, in Chile, this protocol, using wild-type mice. And this is essentially what you see. Did you miss something? Should I or just move ahead? Okay, excellent.
No, it's perfect.
Okay, excellent. So in wild-type mice, that undergo the UCMS, you observe that over the protocol, there is a dramatic reduction in the weight gain, compared to control mice, and in addition there is the fur, the fur deteriorates, dramatically. Those are, indicators of a depressive-like behavior, as you can observe here in this score, based on this table. So the animals, so the protocol, damages, the animals, from a physical, appearance, and weight gain. And this is related, to the impact, on depressive-like behaviors. For example, the sucrose preference, this is, I mean, we are just replicating, previous findings, to establish that the protocol works, in our lab. There is a dramatic, impairment in the sucrose preference test, and an increase in the immobility, revealed in the tail suspension test. We also analyzed, the sociability, using the 3-chamber social interaction. When you evaluate, the preference for a never met before, animal compared, to the empty cage, in mice that have habituated previously, to the box. And then you determine an interaction ratio, and this, is sensitive, to the unpredicted carnivore mice stressors. So, okay, so what happens, with our mice, show me the data, Pablo. So we can observe here, that the overexpressing mice, that are squares, that underwent, the chronic stress, that are the black squares, they do not show, the decrease in weight gain. And in addition, they did not show, the impairments in the fourth state. When we analyze the depressive-like behaviors, again, mice that E3 or expressive mice that underwent the 5 weeks of the UCMS protocol, they did not show the reduction, in sucrose preference. There is no anhedonia elicited in these mice, and the same happens, in the tail suspension test. In the 3-chamber social interaction, well, there is no significant difference, compared, to the control groups. So now we are developing, or we are using, a depression score, and we are exploring, this for the anxiety paradigms, has been, well described, but not necessarily for depression. So we use as a depression score calculating the number of the tests, minus the standardized scores, for each of the ones. We can get like a global portrait, of a behavioral battery, and a given population, and we can use that, to correlate with different variables, rather than the individual ones. So collectively speaking, this depression score can be, related to a pharmacological challenge or to a genetic manipulation. And this is just a reflection of what I previously showed you. We have, increase in the depression score, in the control group, but not in the overexpressing mice. So, now we are exploring, okay, it seems that we have a resilient phenotype here for the chronic stress, of course. These are extremely preliminary data, that I opted to include it, for the scope of this talk. It is known that the chronic stress, impairs, memory, in mice. So the mice that underwent the chronic stress were also tested, in object recognition and object location. This is the establishment of the parameters, again, very preliminary, in the wild-type mice. So there is a reduction in the discrimination index, and the same happens, for the object recognition task. So we perform the same, for the E3 overexpressing mice. I'm sorry. With and without the chronic stress, paradigm, and we can observe, again, this is, very preliminary, but the trend seems to go similar to the other behavioral domains, that there is no impairment, in memory, in mice that have increased expression of E3, tested in the object recognition and in the object location memory. The trend, again, seems to go, in the same direction. So far, I've tried to convince you, that this is a very cool transporter, to study. We went over, data indicated that the increased forebrain expression of this transporter, triggers an increased anxiety-like behavior, increased compulsive/repetitive behaviors, alterations in the NMDA subunit composition, and as per today, we have only demonstrated, in the corticostriatal synapses, functionally, but at molecular level, we know that the changes, are similar in prefrontal cortex and hippocampus as well. We observe, deficit in the long-term extinction. So now, we are actively pursuing, the determination of characterizing the ventromedial prefrontal cortex connected to the BLA, which is, part of the circuit that is involved, in the extinction of fear memories. And we have, these, novel findings about this resilient-like phenotype to chronic stress, based upon, tests in anhedonia, till suspension test, sociability, and the object recognition and location that seems to be intact, in this mouse model, although very preliminary. So I would like to also mention, what are we currently, embracing in the lab. We are analyzing the alterations in the dopaminergic system. As I mentioned, the synaptic, what happens at the synapses in the PFC BLA, alterations in the hippocampal neurotransmission, and on a different, subject, we are manipulating, E3, in oligodendrocyte precursor cells, because it has been shown that during some of the early stages, E3, is highly expressed, and that might determine, the cell fate, of these precursor cells, and might have also some, role, for the myelin formation. Of course, I must thank, to the people who do the job, the collaborators, here in Chile, and worldwide, and the funding agencies, that at this stage, are mainly, national, which essentially translates, to a big thanks, to all Chileans who truly pay taxes. Thank you.
Thank you, Pablo, for this great talk. So it's very interesting. So for the public or for the panelists, if you want to do a question, please write in the preguntas y respuesta or Q&A. But meantime, I have a question, Pablo. So, so the transporter EAAT3 is expressed in postsynaptic. So, what do you think about the mechanism that when glutamate enter to the postsynaptic and cause this this characteristic, this behavior in your model?
Well, there are several alternatives, that we are actively pursuing. One of them is the alterations, in the cysteine levels, because cysteine is the precursor for glutathione, and glutathione is known, to regulate, the activity of the promoter regions of some of the ionotropic glutamate receptor subunits. So perhaps it might have nothing to do, with the glutamate uptake itself, but perhaps, by altering the glutathione levels, that might regulate, NF-kappa beta, and NF-kappa beta, will be the transcription factor involved in the decreased expression of the 2A subunit, which particularly, resembles, like an early development. We are also characterizing the NMDA subunit expression profile across development, because with this adult with picture only at adult, it seems that the receptors did not undergo the normal switch between the 2A and 2C that you see across the development. So we are, analyzing, from early, ages.
I have a question.
Who has a question?
No. Dr. Flores here. So, Pablo, thank you for the great talk. And I was wondering, so just like the SSRIs help with the OCD kind of like behavior in the mice, what do you think will happen with the feed extinction problems?
Well, that's a fantastic question. That's a fantastic question. Ultimately, what is? Today, no one knows how SSRI work. Ultimately, But most of the hints, in terms of what could be the molecular mechanism behind, could be addressed by modulation of the 5H2B receptor that will regulate the serotonergic tone at the cortex, and then regulating what goes from at the orbitostriatal level. So I would like to mimic something from the ventromedial prefrontal cortex, that the SSRI will regulate that tone onto the VLA. But so far we don't know. We have not tested the impact of SSRI on rescuing the feed extinction phenotype yet.
So there are two questions from the public. So Benjamin asked that in the data show that you show that in the control mice at least 1/3 are resilient to the chronic stress. So the question is that there are another mechanism to play in this in the resilience to the chronic stress. That is the question from Benjamin Pizarro. So about another mechanism about resiliences.
Well, okay. So this research, started, in an opposite manner. There was a research project, evaluated glutamatergic alterations in general, and given the availability, of these mice, the mice, underwent the behavioral variety. At this stage, we do not know what's going on, at the molecular level, and in order to link, the consequences of increased E3 function, to the potential candidates that might be, involved in the resilience. So that we have a black box, we have a gap, in the research at this time, to properly regulate, what's going on, with the altered E3 function, and how that could impact, some of the resilience network. So it's currently, investigated.
Okay, here they are. Another one from Paulina Harding. Do you know whether corticosterone has something to do with the alteration of expression or activity of the transporter in chronic stress?
That's a very good question, and I don't have the answer. I don't know if the promoters, the regulatory regions of the glutamate transporters, for this one in particular, and for the family in general, have glucocorticoid receptor responsive elements if they have GRE. I'm not sure if this is a direct link, or it might have something to do, as I previously mentioned, with the regulatory effect of glutathione, over NF-kappa beta, which could be like an indirect, pathway, to regulate.
Okay, and do you know which are the normal mechanisms to regulate the expression of this transporter?
Well, this transporter, is kind of funny because normally, it's like 60%, is located intracellularly, and they go up to the membrane, by demand. So when you have, like a glutamate signal, they, are recruited, to the membrane. So they are readily available, to do their job, but they are not constantly expressed. In addition, it has been demonstrated that the effect of amphetamine, in dopaminergic neurons, depends on E3. So for example, when you administer, amphetamine, the transporter, is internalized, becomes internalized, and that renders, less uptake. So ultimately, the glutamate transmission, is higher, and that is part of the mechanism why amphetamine, induces higher, dopamine release. So it is an interplay, between many things, as usual.
Okay, thank you. I don't know if there are another questions, so from the public or from the panelists. You showed very preliminary data, so the role of this transporter in the memory. So the last slides, I think so. So, what do you think about the mechanism, the role of this transporter in the memory acquisition or memory in this memory paradigm?
That's an excellent question, that I was hoping, to engage, interest, from the audience. I would say that, it's way too preliminary, to take a side. We need to establish, proper experimental setting, to determine, which one, could be.
Okay, I think so. There are no more questions. So thank you so much, Pablo, to attend our symposium. Your talk was really good, really fantastic, with very preliminary data also that always is good. So thank you everyone for the assistance. So I think so it's in general has been a very good morning with these 4 talks. All of them were very, very interesting. So I think so that is the only can say that is thank you very much for the all conferences from all of the speakers, and see you tomorrow at the same time. See, bye, Beb. See you. So. Thank you so much for that end again. So thank you so much, and bye-bye.
Thank you. Bye. Adiós. Gracias.
Bye. Thank you.